Here is the PAM sequence and the two guide. And this guide, the efficiency of this guide has been tested on in vitro produce bovine embryo, because we have a production all the week. And because also the sequence of this region is fully homologous between goat and bovine on the guide, and there is the mutation. So G that is transformed in T is the mutation is here. The ATG of the gene is here. And we use also free single-strand homologous DNA in order to try to have a homologous recombination and to copy past exactly the same mutation. So very briefly, we have done different experiments, eight experiments in total, but in one experiment, we engage 5 donor goat and 6 recipient goat. All these goat was cooled goat, so in order to respect the 3R rules. And so both goats receive a treatment for hormonal treatment, the donor for superovulation and the recipient for to be synchronized with the donor. So the donor, we recovered the zygote oocyte because we, after this treatment of superovulation, at the end, they are inseminated by artificial insemination with frozen sperm, and 2 times. And the day after, we recovered the zygote and the oocyte and slaughtered by flushing the oviduct of the goat. Then this zygote or oocyte, because some are not fecunded, are put in culture during 18 hours in order to have the first stage cleavage. So 15 hours later, we check all the embryo to see those that was fecunded and those that are to develop. And these embryos, these stage embryo, are replaced on a recipient goat with a 3 embryo maximum per goat. After we follow the gestation of the goat, of the recipient goat, by a progesterone test at 21 days by ultrasound scan at 45 and other day. And then we get some founder, and the founder, as I told you, should be crossed with wild-type animal in order to produce F1 animals. So very briefly, we have done 8 experiments. We have manipulated more than 500 embryo, and you see that for this more than 50,000 manipulated embryo, the 157 are cleaved to the 2-cell stage. It's a cleavage weight of around 30%. But you can see that the cleavage weight is very variable according to the different experiments. And these 2 experiments with higher cleavage weight is the 2 experiments where we gave gestation to term. We have some, the other goat have a positive 21, has found, some was found positive for gestation with a progesterone level at 21, but only these 2 experiments, we get 6 recipient goats that are pregnant, that deliver 11 newborn. On this 11 newborn, 8 were found, 8 have some mutation and would be considered as founder animals. And on these 8 founder animals, we get 4 knockouts, 2 female and 2 male. So here is the founder animal with the different mutation. So we get one founder that is very interesting. It's called Caesar. And it was found homozygous for a one base insertion, so giving a frame shift and a knockout allele. And also, remarkably, we have get 3 times this mutation, that is a deletion of 6 bases of 2 amino acids, and actually we are trying to develop 2 goat lines, one from Caesar with the insertion, the one with the del6 mutant, because perhaps in order to assess this small deletion could change the resistance or the sensibility of the goat to prion disease. And so, as I told you, this work has been published last month in veterinary research. So if you want more detail, you will find it in this publication. The second example that I want to present is a project that has been done in the Roslin Institute by Simon Lilico. And it's a practical application of it's an interspecies transmission of the SLIC mutation. So it's to try to investigate thermal tolerance in sheep and to assess the phenotype induced by this mutation in other species than bovine, so in sheep. So this mutation, the SLIC mutation, is mutation of the prolactin receptor gene, and it has been described independently in 3 breed of cow that live in tropical area. So we think that this mutation is a sort of adaptation of the climate and to the heat stress. And briefly, the prolactin receptor is a transmembrane receptor with an extracellular domain, an intracellular domain, and a transmembrane domain. So it acts when it dimerizes, and this amino acid, this tyrosine, is very important because its dimerization is required to activate the JAK-STAT pathway. And all the mutations described in the different breed of cow shrunk the receptor before this tyrosine, and so it certainly repress the JAK-STAT activation. But it's not a knockout allele, it's a new variant. And so and it is dominant, so when the animal carries this mutation, they have a phenotype with a short hair coat and an increased ability to regulate body temperature. So Simon have mimicked exactly the same mutation by also, and Simon, yes, Simon have mimicked the mutation with the same strategy as us, but he obtained a main sheep with homologous recombination. And in goat, we have absolutely no homologous recombination in our, I forget to tell you this, but we have only a non-homologous end joining, so an HG on goat project. We haven't had HDR, and we do not know why. But in the case of Simon, he get many animals with HDR, and this is all the founder gate, and some of them are directly homozygous for the SLIC mutation. So he start to work on this founder, and this is the offspring at birth, and they are quite normal and not different from wild type animals. When they grow, I think this is some picture at 2 or 3 months. You can see, so this is a non-edited animal and a homozygous edited animal. You can see that there is less hair on the head, and the phenotype appears very clearly in spring, in August, 6 months, where you see that there is really less on the edited animal. So, and this is another picture. So if you have also measure the length of the hair at different places, and you see clearly that between the control, there is a big difference. And also, the weight of the fleece, of course, is very reduced. Surprisingly, when this animal enter in winter, the difference disappear. The animal recovered some wool for the winter. So the future work on this animal is to produce F1 and to work on F1 for a more precise phenotype. We will also test this animal for heat tolerance, because in ROSIT facilities, they have all the facility to test the tolerance. And also, he will characterize the wool, the wool of these animals. So briefly, a last example that has been done with INRAE grant. This is a French agency, and this INRAE was managed by Hugues Dardant, a colleague of me, working on seasonality, on seasonal breeding. And so in seasonal breeding, very briefly, we know that the primary signal that are is at the basis of seasonal breeding is the circadian clock, the secretion of melatonin. And we know what happened with this circadian clock. We know also that the endpoint is on the neuron that secrete the gonadotropin, releasing hormone, so the GnRH, to control the secretion of LH and FSH, the gonadotropin. The link between first signal and the end of signal is not known actually. So there is two main candidates, because there is two types of neurons that control the GnRH secretion, the neurons that secrete peptine, and also, and that are well known, but when you not expect, you have no reproduction. And there is also some other neurons that are not be clearly investigated, and these neurons secrete a peptide that is called RFRP. This peptide is encoded by the NPVF genes. So there is no functional study on this NPVF gene. So the project was to try to knock out these genes and to eliminate the peptide in sheep. So we have done 10 experiments, and we get 3 founder sheep with cow allele. So actually, these founder are crossed with wild type in order to have some line to verify the mutation. But I want to present you this project because it's projects really in fundamental research, basic research. But we cannot exclude that the knockout animal will stop to be seasonal. And in that case, you see that it could be very interesting for an agronomical purpose. Just to see you that when you are doing functional genomic farm animals, it's very difficult to, the frontier between fundamental science and applied science is very. So now I want to conclude. There is many things to say on genome editing, so I have the choice to conclude on some like this, but there could be many different ways to conclude. So I think personally that in vivo genome editing represents an essential and revolutionary tools in a number of fields. So for basic research, for agronomy, and for biomedicine, because in order to produce biomedical model. So, for example, Europe wants to see a drastic reduction in the use of antibiotics, and I think that genome editing could make a major contribution to this goal. And also they will approve projects that aim at improving animal health. And there is a nice example from the US on these topics. Is the porcine reproductive and respiratory syndrome that has been published recently. So actually, the problem in Europe that Europe has classified these animals and organisms, plant and animals, as GMO. But actually, the decisions are revised, and you can have some input on this decision. We have proposed a policy paper, RUMIGEN, and last week there is a draft NGT applied to animal from EMSA, from the EFSA, that has been public. So if you want to consult it, it's interesting. And so in the future, I hope that the legislation on this animal relax and that there will be more European call for supporting scientific project. So before ending, I just want to thank many people that have been involved in this project. PI, Jean-Luc Pilote, Simon Lilico, Michael Tonulis, and Carol Charlier are responsible for the different tasks of the package 5 of RUMIGEN. All the staff involved in the embryo transfer and the Alliance Society that was involved in embryo micromanipulation. And I have to find also a member of the free experimental facility from INRAE, because this project involved free experimental facility. I thank you all, and I to answer your question.
Right. Great. Taking time. Next question from the audience. I have a question. I'm just wondering, have you guys done any of the deep sequencing for those gene editing animal to see if these got RNA or target other genome?
No. You can check to predict an off-target site, but in our case, we cannot. It has no sense to sequence this animal because we do not know the parent. Because we use 5 donor goats, and then the embryo are pulled to be manipulated very rapidly. Alliance and Aurélie manipulate 15 embryo in 15 minutes. So we cannot check if they are fecunded or not, and we do not know from which goat they come. So it's very difficult to find the off-target by whole genome sequencing. Very difficult. This is a project on task bovine, and on bovine, this is exactly the same guide that has been used on bovine embryos and the guide that we use for the goat. It is also genome editing of prion gene, but we stop the development at the blastocyst stage. It is blastocysts that are sequenced. So we will see if there is many off-target.
So very interesting. 2 questions about the goats. So first, said that you didn't have any HDRs.
Yeah.
Is there a technical difference between what Simon does and what you did in terms of the way you deliver?
I think it depends on the target.
Okay. So it's not technical.
Yeah.
It's not. So does he use electroporation, use injection?
The target that we want, the prion genes, have a specific structure that do not hybridize with a single DNA. We have changed the DNA in between. The 5 first experiment was done with a single-strand DNA, and then the 3 other with another single-strand DNA. But in both cases, we haven't seen any HDR.
And the aging phenotype, do you know how old are these animals now? what's the phenotype of this animal compared to
But
wild
we
type?
haven't phenotype because to phenotype them, we have to kill them because to take their brain and to test if they are sensitive or resistant to prion. So, and it is founder animal. And for example, Caesar that we found homozygous in the skin of the. We have genotype on the DNA from the skin, but now we have some F1. We've seen that the germinal lineage is mosaic because its kids, most of the kids have the +1 insertion, but it was a +A that were found on the DNA of the skin, and now in some kids there is some +C. And there is also a very few wild-type animals. So this goat is mosaic. So that's why we want to make a line to genome and to phenotype. Normally, they are predicted to be resistant. The more interesting is the adverse, the other phenotype.
Okay, it's a interesting time. Thanks very much, Eric, for your talk.